It's my pleasure to introduce Alicia Pickrell, who's at the Virginia, Virginia Tech in the Department of Neuroscience or School of Neuroscience. Um, so Alicia got her PhD down at the University of Miami with Carlos Moraes, who's really big into treating mitochondrial diseases and has done some really cool work in that area. She then, after getting her PhD, moved on to the NINDS under Richard Ewell, where she spent a number of years really digging into mitophagy, so mitochondrial quality control pathways. And she did some really nice, highly cited work in that area. And that's actually where I met Alicia, because we actually have a paper together. And then, as I transitioned over to faculty, she stayed on for a couple more years. And then in 2017, actually got a position of her own at Virginia Tech, and where she has been since. And she's here to talk about mitochondrial dysfunction and in disease. And with that, I will turn it over to you.
Thank you, Scott, for the nice introduction. So as Scott mentioned, I started mitochondrial research when I was a PhD student and have continued to maintain interest as a postdoctoral fellow and as well as starting my own laboratory. And so I've really been fascinated with mechanisms by which mitochondrial dysfunction contributes to disease. So really, I've broken my talk down into 3 different sections. So the first section, I'm going to talk about how classically mitochondrial dysfunction is seen as a trigger of causing cell death. And I'm going to focus on defects in mitophagy or the removal of damaged mitochondria and kind of discuss the work that I collaborated with Dr. Kennedy. And then I'm going to move into how these same proteins that are involved in mitophagy are also involved in causing problems in cell cycle progression. So it kind of seems a little bit different. First part, we're going to talk a little bit about death. Second part, we're going to talk about cell cycle progression. And then finally, I'm going to discuss unpublished data from my laboratory and hope to get some feedback on our studies into studying mechanisms of mitosis and how we're kind of setting up the laboratory to study the physiological consequences of disrupted cell division and dysfunctional mitochondria. So I'd like to remind you that mitochondria are the powerhouse of the cell. So this double membrane organelle, they're found in every cell of the body except for red blood cells. And importantly, they provide energy for the cell by generating ATP. So you can see that first line with the bullet point, ATP generation. And some cells are really highly reliant on ATP to provide this energy and really rely on their mitochondria. So neurons, for example, they need a lot of ATP and they're highly reliant on oxidative phosphorylation. So they really can't utilize glycolysis as other cell types may be able to get their energy elsewhere. And you can see that I've also listed that mitochondria are also important for other different parts of functions that are required for cell viability. So you can see calcium regulation. Mitochondria are great at buffering calcium when there's excess and also utilizing that calcium. You can see they trigger cell death. They also important for lipid metabolism, reactive oxygen species handling. So a lot of the different enzymes to detoxify reactive oxygen species can be found in the mitochondria, and many others. And not only that, but I like to bring up this little cartoon that I stole from somewhere on Google, but you can see that mitochondrial dysfunction is implicated in a wide variety of human health maladies. So whether mitochondrial dysfunction is the primary contributor or the secondary contributor, you can see that it's been implicated in many different types of diseases, not just in the nervous system that have neurons that are highly energetic, but you can see quite a long list here on this little figure on the left. And just a reminder about how it makes this currency for the cell, or ATP. We have this inside of each of our mitochondria. We have a series of complexes. So mitochondria make energy through these protein complexes in the inner membrane. And these particular complexes perform a series of redox reactions that are able to shuttle protons into the inner membrane space to generate this proton gradient that can then be utilized by complex 5, or this ATPase synthase. And this will be able to have the cell from ADP to generate this ATP currency. And you can see that this process also utilizes the consumption of oxygen as well. And because I was talking about how neurons really can't rely on other ways to utilize glycolysis to make ATP and get that energy that they need, you can definitely see a lot of different mitochondrial defects associated with neurodegenerative diseases. So here on the left-hand side, you can see a little picture of the human brain, and then different regions of the brain where you can see common neurodegenerative diseases with different regions of the brain that are affected, where neurons are dying. And in these different diseases, in that last slide with oxidative phosphorylation, there are different complexes that seem to be affected in each disease. So for example, in Alzheimer's disease, complex 4 seems to be affected in this OXPHOS pathway, whereas in Parkinson's disease, complex 1 seems to be more affected. And so in all these diseases, you can find that mitochondria are dysfunctional, and it's early in the disease, so it's an early event. And different neurons seem to be susceptible in each one of these diseases. And I'm going to focus on mitochondrial dysfunction associated with Parkinson's disease. So if we look here in Parkinson's disease, I have a little cartoon of an individual. And approximately 1 million Americans are affected with this age-associated disease. And that's more than those combined affected by other neurodegenerative diseases, such as ALS or multiple sclerosis or muscle dystrophy. And there are 4 cardinal signs to this disease. One is bradykinesia, one's rigidity, the other one is postural instability and tremors. And bradykinesia just means the slowness of movement. And this is the most common motor deteriorating neurodegenerative disease. And most people have this form of disease where it's called sporadic. So 90% of these individuals with Parkinson's disease, they're categorized as sporadic, meaning we don't know what caused their particular disease. And then the other forms are monogenic or genetic, in which we can see in their DNA, there's been some gene mutation that has made them either more susceptible or provided an early onset form of the disease. And so symptoms really appear when approximately 50% or more dopaminergic neurons in the substantia nigra are affected and degenerate. And the loss of these particular neurons actually cause a depletion of the dopamine in the striatum. And so there's no cure. So you can see here, there's a picture of an individual with Parkinson's disease, and you can see that these neurons are actually colored in the human. And so you can see them here, but that they're absent in this individual. And so since there's no cure for this disease, there's only symptomatic relief for patients. And one of the pathological identifiers of this disease is the accumulation of aggregated alpha-synuclein and ubiquitin positive inclusion. So you can see here, this Lewy body made up of these aggregated proteins. However, we do find, so here we have some genes that have been found to cause early onset forms of Parkinson's disease. And we can see that there are 2 genes, PINK1 and PARKIN, or PARK2 and PARK6, that encode the proteins PARKIN and PINK1. And these are recessive mutations. So that means that an individual will have 2 of their alleles that have particular mutations that cause a loss of function of the particular protein, and that causes early onset Parkinson's disease. And I'm going to focus on talking about these 2 genes. So you can see that I have a little cartoon here, and over the last 14 years, you can see that the Yule Lab and others have really contributed to understanding the mechanistic aspects of this particular 2 proteins. So you can see that PINK1 and Parkin actually were first found to be genetically linked through work in Drosophila. And then a lot of work in cell culture and in particularly cancerous cell lines have really worked out the mechanistic detail of this particular pathway. So we can see that PINK1, if we look here in the middle of the chart, PINK1 is accumulating on this damaged mitochondria, but under normal conditions, PINK1 is constitutively imported inside the mitochondria because it has a mitochondrial targeting sequence. And then it is processed inside the mitochondria and then released and then degraded by the ubiquitin proteasome system. So in a normal cell that has healthy mitochondria, we really don't see a lot of PINK1 present because it's constantly being degraded on the protein level. However, if a mitochondria becomes damaged in some way, and you can see a little picture of particular damages, loss of membrane potential or increased reactive oxygen species or mitochondrial DNA mutations, we can see that PINK1 accumulates on the outer mitochondrial membrane. And that's because import is blocked. When that happens, PINK1, it's a kinase, so it can phosphorylate different substrates, and the substrates that's around the PINK1, whenever it's on that outer mitochondrial membrane stuck, it will phosphorylate ubiquitin chains on other mitochondrial proteins that are stuck on the outer membrane and Parkin itself. So Parkin is normally cytosolic and it's an E3 ubiquitin ligase, but with this particular event, it's recruited to the outside of the mitochondria. It starts ubiquitinating a lot of other particular proteins, and these events cause the autophagophore to form around that damaged mitochondria and send it to the lysosome for degradation. So the prevailing thought is that if you have particular individuals that don't have PINK1 or don't have Parkin, then you're going to accumulate dysfunctional mitochondria because now we can't clean them up or clear them by the lysosome. And that's going to cause certain neurons that are highly reliant on their mitochondria to have energy to degenerate, leading to disease. And this would probably happen over some amount of time. However, when I joined Richard's lab, a lot of people had tried to make different models of PINK1 and Parkin, particularly different germline knockout mice, and they really didn't see any sort of problem with these mice. And so we can see that in the PINK1 knockout mice and Parkin knockout mice, different strains were made, but they really didn't see anything happening. Normal lifespan, no discernible phenotypes, no really Parkinsonian type symptoms. And so in my postdoctoral work, we utilized this mouse model of mitochondrial aging to test whether endogenous Parkin was really necessary for the survival of dopaminergic neurons. And so I'm going to just talk about what mitochondrial DNA is. I'm sure you already have heard what it is, but just as a reminder, you can see that mitochondrial DNA, it's very important. We have many mitochondria inside of our cell, and not only that, each particular mitochondria has tons of mitochondrial DNA genomes. So you can see this little picture depicted in this cartoon. And so there's multiple copies of mitochondrial DNA within a mitochondria and multiple mitochondria within a cell. And it's this 16.5 kb genome where these molecules are important for encoding 13 proteins or polypeptides that are membrane spanning for complexes 1, 3, 4, and 5. And so we can imagine that if we have some sort of problem in our mitochondrial DNA, we can have some sort of mitochondrial deficit. So we can see here I like this cartoon from this review. So we can have different levels of heteroplasmy in different tissues, in different cell types. So we can see that because we have multiple copies of mitochondrial DNA, we can have a mix of healthy, non-mutated mitochondrial DNA and mutant copies of mitochondrial DNA. And we can see that perhaps if we have too many mitochondrial DNA that are mutated, that's going to then lead to some sort of mitochondrial defect, especially in those complexes where mitochondrial DNA are very important. So complexes 1, complexes 3, complexes 4, or affecting ATP generation. And so there were already papers published too that had really shown that if we look at sporadic Parkinson's disease patients or even aged human healthy patients, we can see that in their substantia nigra, they have large scale deletions or large portions of their mitochondrial DNA missing. And these deleted molecules reach high levels of heteroplasmy. And whenever they also looked at these particular dopaminergic neurons, these two groups also correlated that particular finding with a loss of mitochondrial function when performing some in situ enzymatic activity assays. So they were able to show that, having these large scale deletions and mitochondrial dysfunction really happens in sporadic PD and even aged healthy humans in these neurons that degenerate. So going back to this slide, we were going to use this mouse model of mitochondrial aging, and you can see that it has a particular point mutation in this gene called polymerase gamma. And polymerase gamma is really important because it replicates the mitochondrial DNA, and by having this particular point mutation, every time the mitochondrial DNA replicates, if a nucleotide is misincorporated during the replication process, there's no repair. And so over time, these animals actually have a decline in mitochondrial function. They have premature aging phenotypes, and they really have a shortened lifespan of about 1.5 years. And you can see that these guys looked like they're in pretty bad shape. But a previous publication already showed that even though these mutator mice, they had a loss of mitochondrial function in the brains of different regions that were looked at in this particular paper, they also showed that there wasn't really any marked neurodegeneration. And so we can see here that they looked at some brain slices and they were able to do enzymatic activity on these particular brain slices, looking at complex 4, which is the brown deposit, and looking at also complex 2, which is the blue deposited color. And we can see that if we look in the mutator mice at the 46-week time point here in this column, we can see that our wild type mice have a lot of brown, but unfortunately, you can see that our mutator mice have a lot of blue, and blue is not encoded by the mitochondrial DNA, so it shouldn't be affected. So these animals with age accumulate tons of mutations, and we can see that it's going to affect the mitochondrial function of these particular mice with age. But really, these brains look pretty good for mouse brains. There's not really a lot of degeneration visibly looking at this gross neuroanatomy. So when we looked and analyzed these particular mutator mice that either had or did not have Parkin, we saw that whenever we looked at these aged mice around the 48 to 52 week time point, which was when that previous publication was showing that a lot of mitochondria were dysfunctional in this particular mouse model, we can see that in B, when quantified, we see a loss of dopaminergic neurons. And we also see that this is progressive. So I don't really have time to show all the data, but we see that these animals at younger ages really didn't have any sort of neurodegeneration, and that we saw that this was definitely dependent on L-DOPA, which is a drug that they normally give PD patients to provide symptomatic relief. We also see here that with this data, we collaborated with Wade Harper's group, and we detected phosphorylated ubiquitin. So one thing, when we were doing this project at the time, we weren't really able to detect mitophagy. And so we kind of did it in a way that was, we knew that PINK1 would phosphorylate ubiquitin, and it seems to be so far the only kinase that does so. And so we took the cortical lysates of our particular mice at this age time point, and then we went ahead and pulled down all the ubiquitin chain proteins using affinity tagged tandem UB binding domains. And then in collaboration with his group, they performed absolute quantification of proteomics. So we could quantify the amount of phosphorylated ubiquitin. And so we see that in our mutator animals, it looks like PINK1's doing something because we see an increase in this phospho-UB signal here. And we can see that when we looked at the striatum of these old mice, that we were able to see that looking at either complex 1 and A, complex 3 and B, or complex 4 and C, that we were able to see that there were some deficits that were more severe in the mutator Parkin knockout mice, at least for complex 1 and 3. Complex 4 was equally severe. We can see that in C. And we didn't see any detectable mitochondrial dysfunction in our other groups at 3 months of age, all 4 groups. So conclusions for part 1 is that dopaminergic neurons, they degenerate in mutator mice that systemically lack endogenous Parkin, and that phosphorylated ubiquitin is increased in the brains after mitochondrial stress as a proxy to say that mitophagy is happening, and that loss of Parkin exacerbated mitochondrial dysfunction in these neurons. And I didn't have time to go over the data today, but Dr. Kennedy really helped us out with a lot of the sequencing to start to characterize those particular mutations and quantify them. And so here in the second part, I'm going to switch gears and talk about how these same proteins that seem to be important for keeping these particular neurons from living or dying have a different role in cell cycle progression. And so just a little reminder about cell cycle. So we have this little picture on the left, and we also have a picture of bad things that can happen if cell cycle was arrested. So if we first talk about the stuff on the left, so cell cycle, it takes place as cells are going to grow and divide, right? So when we think about neurons, we really don't have to think about that because they're post-mitotic cells. But most of the time, the cell is actually in a growth phase. So this phase is called interphase, and it's going to grow. And then eventually during S phase, we're going to replicate the chromosomes. And then finally, we're going to have another growth phase before the cell prepares for division. And we can think of cell division as 2 processes. So mainly characterized by this DNA replication phase, and then also by the segregation of these chromosomes into 2 separate cells. And we can see here that they've got all these little, this diagram that I took from this review, we see that all these little proteins, so it's all these cyclin-dependent kinases are really steady state in the cell, and they are important for progressing these different stages of the cell cycle. And so these cyclin proteins actually rise and fall and are regulated by the ubiquitin proteasome system to go ahead and bind to these CDK1 proteins to activate these different phases. And you can see on the right-hand side that if you have problems with mitosis, so during this time when we're going to segregate the genetic material and then split it into 2 cells, we can have a whole bunch of bad things happen. So we can see that you can die. We can see that the cell might have too much DNA, and so that it's multinucleated or binucleated with too much genetic material. We can also see that maybe not only that, but if we have too much or too little genetic material, we can die again, or we can senesce, or we can perhaps with other gene mutations proliferate too much. So we can see that there's definitely consequences to being arrested. And there were a lot of particular papers that were coming out showing that these PD-related genes also may have some role in cancer. And so the literature really suggested that there were a lot of different deletions or different types of mutations in PINK1 and Parkin. And you can see the different research papers that I've listed on the left hand of the slide that really show that they're involved in maybe glioblastomas or colorectal cancer. And you can see that I have a little figure that I took from one of these particular papers. And you can see in A that they looked at pretty much 10,000 different types of tumors across 28 different cancer types. And then they categorized whether they had some sort of deletion in Parkin or some sort of a large portion was deleted, or maybe nothing happened at all. So that's our little graph in A. And then we can see that in B, we have that the mRNA level. So they were kind of looking at these same particular samples, and they wanted to see that in these different cancer types, was the mRNA up or was the mRNA down? And you can really see that there was either no change or the mRNA was actually impacted by these mutations and decreased. So there was less Parkin in the cell. But one thing was that Parkin, in the beginning of my talk, I showed that really it needs PINK1 to do some stuff, right? It's just hanging out in the cytosol, and you really need to activate it with the PINK1 to phosphorylate it. And not only that, but PINK1 is always at the mitochondria. So we know it has that mitochondrial targeting sequence, always getting in there, always getting degraded. And so really, how is this working, right? And so one of the things that we did in my laboratory is we first set out to do some genetic screens. So because the PINK1 Parkin pathway was elucidated in Drosophila, we collaborated with Nikos at Biogen and Dr. Lena Nee at Virginia Tech to help us set up some Drosophila screens to see if PINK1 and Parkin really genetically interacted with the cell cycle in any way. And we were able to follow two outcomes, survival and the thorax indentation phenotype. So in the flies, PINK1 and Parkin flies, they do have dopaminergic neurodegeneration, but they also have this, you can see in this little picture that I took, that they have this thorax indentation phenotype. And so this particular phenotype is because during development, the flight wing and the thorax, they develop from adult muscle precursors that and a highly proliferative stem cell pool. And so we evaluated PINK1 and Parkin flies immediately after they came out and hatched, so occlusion. And we looked to see if we knock down different cell cycle proteins, whether that had some sort of effect on this particular thorax indentation phenotype. And we can see that here in E and F, that we were able to rescue the phenotype of these particular flies. So counting 200 to 300 flies per different crossing, we can see that we have this rescue. And so I'm just showing CDK1, which is important for progression through mitosis, but we also see this in CDK2 and also another particular regulatory protein that's important for cell cycle progression. And this was interesting. This kind of confirmed some of these findings that maybe there's some meat to this story that PINK1 and Parkin are important for cell cycle stuff. But we really had those same problems in our mind that, you know, PINK1's at the mitochondria, Parkin's not doing anything without PINK1. So really what could be happening? And one thing is that reading the literature and looking back, we saw that there's really a protein that connects both mitophagy and cell cycle. And it's actually this protein called TBK1 or tank binding kinase 1. And this particular protein was really well studied and first described in innate immunity. So it's important for phosphorylating transcription factors that go to the nucleus to stimulate an interferon response whenever you have viruses or bacteria or some sort of innate immune stimuli. And the crystal structure had really been elucidated at this point, and we can see that with this little cartoon that you need some sort of adapter to bind to a particular TBK1 monomer. And then this binding will go ahead, activate the kinase domain through this trans autophosphorylation. And then the kinase can go ahead and activate its different substrates downstream. So we can see that actually 3 different groups independently, Dr. Richard Yule, Dr. Eric Holzbauer at UPenn, and Dr. Wade Harper, all had papers around the same exact time showing that TBK1, this protein involved in innate immunity, was also involved and important for mitophagy. So basically these papers all showed that there's a little cartoon, but I even have a little immunofluorescence experiment here, that when you had mitophagy happening with this PINK1 Parkin pathway, they saw that TBK1, this kinase, was recruited there, and it helped phosphorylate different adapters to facilitate the autophagoform forming around the damaged mitochondria. And if you didn't have PINK1 and you didn't have Parkin, they didn't really see TBK1 there present. So yeah, you can see here that phospho-TBK1, or that activated form, is on the mitochondria with the Parkin, but you don't see it in untreated cells. And like I said, PINK1 and Parkin are required. And so this on the top is our data, but 2 other papers had shown that TBK1 is involved in mitosis and cell cycle progression. So if you knock out TBK1, you see that the cells don't really divide as well. You don't have as many cells, the cell growth slowed, and that you have an accumulation of multinucleated cells. And we can see here that phospho-TBK1 is on the centrosome, basically in all stages of mitosis. And we can also see that we have the presence of phosphorylated TBK1, so the active form, during mitosis, but you really can't see it in asynchronous cells because we really only have maybe like 5% of the cells actively dividing in mitosis at that stage. But we can see that when we sync the cells up, we can watch that signal where we have activated TBK1. And so we then thought about our fly data and we thought, well, maybe this is an explanation for how maybe there's some involvement between, or some connection between, these mitophagy proteins and cell cycle. And so here we performed an experiment where we looked to see if TBK1 was either on the centrosomes or during mitochondrial damage, we saw it on the mitochondria. And so we synced all these cells to the G1S phase. And then whenever we had these cells synced up, we looked to see where these particular phospho-TBK1 signals were. And so we can see that whether we had Parkin or not, if we just release these cells normally, we saw that all the time the phospho-TBK1 signal was on the centrosomes. However, we see that whenever we have oligomycin, antimycin, so if we have some poisons that damage the mitochondria pharmacologically, we can see that the TBK1, whenever we go ahead, we sync up these cells, and then we release them in this mitochondrial damaging media, we see that all the time it's on the mitochondria. And this definitely has an effect on the cells. So we can see that as we're following these cells, we're seeing that a lot of them are able to go through mitosis at 9 hours later. However, whenever we have active mitophagy with the Parkin, but no, we can see that we decrease the percentage of mitotic cells as opposed to not having any Parkin. But mitochondrial DNA damaging agents, they've already been shown that they affect cell growth. So if we have them in the media, it's not really surprising, even though we see a significant difference here. So to further test this idea that maybe TBK1 in one place is going to cause an effect that would sequester it away from the centrosomes where it's needed to progress cell cycle, we actually performed this experiment where we used a FRB, tethered Fis1, and FKB TBK1. So we can use this rapalog induced dimerization system. I'm sorry, I don't know why my slides keep progressing forward. I think when I practiced, I must have practiced a lot faster. But anyway, we can see that whenever we go ahead and then we look at this particular rapalog-induced dimerization system, we can artificially tether with the addition of rapalog TBK1 to the mitochondria without hurting the mitochondria. So we can use this mitotracker red, which is responsive to the membrane potential. And we can see that our TBK1, we can tether it artificially to the mitochondria, adding rapalog in these TBK1 knockout cells. And we can see that that decreases mitosis. So when we have all of this TBK1 that's over there on the mitochondria, these mitochondria are healthy, we can see that we have this sequestration. So this second part, basically, we were able to show that there is some sort of genetic link between PINK1 and Parkin and cell cycle progression, and that we believe that it's due to this protein called TBK1 being in one area versus another. So we believe that, TBK1, it's active during mitophagy, it's also active during mitosis, and that when the cell needs to divide, we can't really have any TBK1 there, that it's somewhere else, like cleaning up the damaged mitochondria, that's going to cause cell cycle to be blocked. So I'm finally going to start talking about some of my unpublished work from my laboratory, and I have to really thank the people that are doing the work. So I'm going to talk about a little bit about mitosis and where we've kind of tried to figure out some of these questions. And this was all spearheaded by graduate student Swagatika Paul. And so she's really helped uncover the mechanistic detail about TBK1's role in mitosis. So I already talked about this particular protein, and it seems that it has a lot of different hats, that it can work in innate immunity, it can work in mitophagy, and it works in mitosis. But we really have no idea what it's doing there whenever it's on the centrosomes. So there's been a couple substrates that have been described in the literature, but what is it doing there and what is it phosphorylating? We don't know the whole landscape. And we also don't know how it's activated. So we talked about how it needs some sort of adapter, right, to dimerize and activate itself. But we don't really know what that is. So you can see over on the other cartoons that we kind of know, but we have no idea. And so really what Swagatika did was that first she started to screen the potential adapters. So there's a lot of redundancy. So if you looked back at my last slide, you can see that like proteins like optineurin or NDP52, they also can work in innate immune contexts. And so there's a lot of redundancy between some of these particular proteins and they're shared. So she started screening all the known TBK1 adapters. And so we can just look and focus on C and F. And we can kind of see that we see NAP1 in our cell lines, that we have went ahead and made a stable knockdown cell line. But we see that in this cell line, when we have this mitosis, these cells synchronized in mitosis, we don't have activated TBK1, which isn't true for any of the other adapters that we kind of looked at, either in knockdown stable cells or in knockout cells. So really that made us think that this is the guy, or this could be the guy. And so that got us excited. And NAP1 never has been described to have any role in cell cycle or mitosis or anything. So we were excited to see if this really held up. And so we made some HeLa knockout lines, and we see that in A, we see the same thing. So we synchronize our cells, and we never really see any sort of phospho-TBK1 happening. And we see that also when we look at the staining on the centrosomes, we see less staining. And these cell cycle defects, we see mitotic cells, the number of mitotic cells decrease, as well as multinucleated cells also be affected. And they kind of look similar to the TBK1 knockout, which we would expect if they're in the same pathway. So we felt good about these results, that, you know, they kind of phenocopy each other. And so it looks like activation of TBK1 is also important. When we lose NAP1, we seem to lose that activated TBK1. And so then we went ahead and we see that mitosis is abnormal. And these are just some pictures of the defects in our particular NAP1 knockout cells. And we can see that they have multipolar spindles, we have monopolar spindles, and we quantified all of these defects that I don't have time to show you here. But we can see that when we tried to make some knockout cell lines or stable knockdown cell lines and a diploid line, so these were in HeLa cells, we were unsuccessful. We failed multiple times. So what we went ahead and did is we went ahead and really leaned on our collaborator, Dr. Alban Orderio at Sloan Kettering, and his postdoc Kiham Nam, who helped us make this inducible cell line in a diploid DLD1 line to help degrade NAP1. So this is called the DTAG system, and where we can knock in this particular FKBP variant into our loci of interest. So we can see that here. And this particular knock-in fusion protein then is responsive to this chemical called DTAG, which will target it to an E3 ligase for degradation. So we can see that in our cell line, we see that we can get NAP1 to degrade either with 20 hours or with a shorter time period, and that we lose that phospho-TBK1 signal again during mitosis when we have those cells synchronized. So we can see that looking at this more normal cell line, we can see the same results that I showed you before. So we see that the mitotic index is significantly decreased. We see a lot of abnormal cells again, a lot of abnormal cells. And we did this over 20 hours, which is one cell division period for these DLD1 cells. And we can see that when characterizing all of these defects with cells that were treated with this DTAG degrading the NAP1, we see a whole bunch of different defects, like those monopolar spindles that we saw in the HeLa cells. We see too many spindles like we saw in the HeLa cells. And we have a little graph here or pie chart quantifying those. We also see that, you know, we thought that it was only involved in mitosis, but it looks like that it also extends to cytokinesis. So when we look at cells that are dividing and they're exiting mitosis and starting the division process, we see a ton of abnormal cytokinetic cells as well. And these are just little pictures of the examples that we see when we treat with that particular drug degrading the NAP1. So we can see that we have unfinished cytokinesis, and we also have too many cells. So here's 3 cells trying to divide together. And these cells, we have increased cell death. So after this division period, a lot of these poor cells that are doing this, they're going to die. I told you that we need this binding. The crystal structure data suggested we need this binding to see anything happen with this activation. And so we did a bunch of IPs, and we see that that's true. So if we look and we take cells and we transfect GFP NAP1, pull down that GFP, then we see more activated TBK1 than whether it's in mitosis versus when it's just asynchronous cells. So we can see that here in our IP. And whenever we have a mutant NAP1, which is deleting the TBK1 binding domain, we really don't see any TBK1 binding. And we also see this in an endogenous IP. So if we take those DLD1 cells, we pull down TBK1, we see more NAP1 binding during mitosis. And finally, we see that it's on the centrosomes. So if we go ahead and we look at our particular cells and we transfect them to look at EGFP-NAP1, we go ahead and we see that on the centrosomes in the vicinity of the activated TBK1. So we see what about the substrates? And this has been somewhat of a work in progress that we're still working towards. But we started with our collaborators at Sloan Kettering and Albon's help to design a phosphoproteomic analysis of the substrates to try to explain why we have so many defects in our TBK1 knockout HeLa cells, in our NAP1 knockout HeLa cells, and now our new cell line. So we took our different cells, we synchronized them into mitosis, and then we went ahead and we had either wild type cells, TBK1 knockout cells, TBK1 knockout cells treated with a specific TBK1 pharmacological inhibitor, or just wild type knockout cells treated with a TBK1 inhibitor. So that would allow us to hopefully identify substrates downstream of TBK1 by having these 4 different groups. So that way we wouldn't detect phosphorylated sites that maybe were only due to genetic manipulation of the CRISPR cells, or maybe only due to the pharmacological inhibition of TBK1. And so we got this huge, nice volcano plot. So we identified 63 sites in 56 different proteins. And you can see that some of these sites, we kind of already knew that TBK1 was phosphorylating it. So we can see this is actually stands for P62. So we know that in mitophagy, TBK1 phosphorylates P62. We also see that as well with another protein here, TAX1BP1, right here. So we see a lot of different colors in our graph. And basically what we found is that not only do we have TBK1 direct substrates, but we probably have other kinases that are being affected by the manipulation of TBK1. And we're currently working through this list, and we can kind of discuss our progress if people have any questions. But one thing that was nice about this huge list that we now have that we're going through is we're trying to figure out that we see that when we look at this list, a lot of them are involved in chromosome segregation and mitotic cell cycle, which is what we would expect. And so really this indicates that affecting TBK1 is going to affect cell cycle progression. And so bringing it all back, because it seems like we took a road detour, one thing that we did find that was interesting is that I already told you we kind of screened all the adapters. We really didn't see anything except this NAP1 hit. But if we look at our volcano plot, we see all of these guys popping up that are involved in mitophagy. So TAX1BP1, we see this is NDP52, this CalicoCo2. We see P62, which is this SQSTM1. And so why are you there, right? Why are you by the centrosomes able to be phosphorylated? And if you are, is that some sort of protective mechanism to maybe not clean up some damaged mitochondria during cell division, where maybe mitochondrial inheritance between daughters could be perturbed? So if we always, our past publications thought there was TBK1 being sequestered away from damaged mitochondria, or maybe at the damaged mitochondria and being sequestered from the centrosomes. But maybe that's not just the whole story. Maybe there's other regulatory proteins in place. So just my last couple slides, I'm just going to talk about how we're trying to figure out, so we have these nice mechanistic stories, but how is that physiologically relevant, right? And then I was talking about how we were studying mitophagy and post-mitotic neurons. But one thing was that after that paper was published, a lot of labs started looking at these particular mice that they generated to look at mitophagy. So these mitophagy reporter mice using different fluorophores to go ahead, and these fluorophore properties are going to change whether the mitochondria is in the cytosol or the mitochondria is inside the lysosome being degraded. And so we can see here data from Torin Finkel's publication that he had this mitokaima mouse. So he is looking at neural stem cells, and they are actively going just basally, a lot of mitophagy is happening. And this is super interesting because people normally think stem cells are very glycolytic and don't really use their mitochondria very much. And so what is the physiological relevance of, having mitophagy in dividing cells, and trying to understand maybe how our mechanistic work really plays into maybe the physiological consequences of, not being able to divide, or, not being able to clean up your mitochondria. So this is just some preliminary data showing that adherent neural stem cells, they have activated TBK1 during mitosis. So we can see that we have a stem cell marker, they're actively dividing, and that we can see that we have our phospho-TBK1 guy that's active on the centrosomes of these neural stem cells, and that as we differentiate these cells into glia or astrocytes, we see that that is going away or decreasing. So we can see that basically we kind of want to know why, if we have mitophagy or we don't have any mitophagy in this particular neural stem cell context, where we can see that there's a lot of mitophagy going on, what does it have to affect maybe the neural stem cell stemminess or cell division, or even differentiation potential later on down the line, either into glia or into astrocytes. So conclusions from part 3 is that NAP1 is required for cell division, activates TBK1 during mitosis at the centrosomes, and that, TBK1 substrates during mitosis, they affect other cell division kinases, as well as we have some direct TBK1 substrates that we are currently looking at. And overall conclusions of the talk is that we have dysfunctional mitochondria, and that can cause cell death, but we can also affect other processes in the cell as well. So even though we have less energy in a particular cell, doesn't always mean that it's going to die. And then the other take-home message is that, proteins that we have a particular function for, that this is an innate immune protein, or this is a mitophagy protein, or this is a mitosis protein, maybe we need to start thinking about how those particular proteins might do other stuff in other cellular contexts that we really never thought about before. So I really have to thank the people in my laboratory. I highlighted the people that have been working on the last project. I really have to thank Swagatika, Sahita, Nicole, and Lila, who is our undergrad, who's been great. All of our collaborators for these 3 separate projects, I kind of put them in different colors. And we can't do as much work as we do without these people helping us. So thank you very much. And then people who give us some money. So I'll take any questions if there are any, and here's your QR code that you need.
Thank you for the fascinating talk. It's really nice to see all the stuff that you've done since we've last interacted. Just want to open this up for questions if anybody has any. I have one, but I will let other go first. So, while people are thinking about questions, is the effect of the TRB is associated with the centromere? Is it a binary thing? Is it, it's all there or it's all somewhere else? Mm-hmm. Or is it like if you start to into the mitochondria, like if you were to titrate the amount of dysfunction, does it start to go away? Because I could think that could have implications on how it or what it's doing or why it's doing it. Mm-hmm. If it's all in the centromere and it's saying, hey, keep on going, and then all of a sudden it goes away, and then that acts as kind of like a checkpoint protein. Whereas if it's kind of a continuum, then that would suggest that it's not a hard break. It's something else going on.
Yeah, so that's actually a good question. And with the mitophagy field, we are still struggling over a lot of the things that we do. We either damage all the mitochondria or we damage none of the mitochondria. And we haven't really found a happy medium where we see maybe some mitochondria damaged and we see that, you know, pink one and PAR can go there. So Richard did have a study where he was looking at misfolded proteins, and he kind of showed that a little bit in this JCB study where you can have like maybe some activation here, but not somewhere else. So in the field, a lot of people use all these drugs to just damage everybody to see this particular response. I think that things are getting a little bit better. So a lot of the work coming out from Wade Harper's lab is showing and doing more nuanced studies where they're able to detect phosphoproteomics and phosphate changes due to pink one to get to what you're kind of asking, like this titrated thing. But I think with mitosis, it's probably all there. So I think that this particular protein's all there during mitosis, because mitosis is more like an all or nothing thing, or I'm stuck type thing. But
yeah, I'm just wondering if there was like a threshold effect, like if it reaches a certain level on the centromere, then it's like, okay, we have enough signal, we can progress on. Whereas otherwise, it's saying no, you know, we don't have enough. Something's gone wrong. It's not where we think it should be. Stop.
Yeah. And we really don't have a good answer for that. There was a paper showing that maybe autophagy works like that. So there was a mole cell paper kind of showing that you really can't get any autophagy during mitosis. And they really they kind of linked that to CDK1, the major mitotic protein to progress mitosis. So it might be a similar mechanism. Because in my mind, I think, why would I clean up stuff if I've got to divide? If I'm actively cleaning up, I could clean up too much, right? And then my little friends don't get any mitochondria, or maybe, you know, I don't do a good job. I think it's probably kind of risky. And then also mitosis is very bioenergetically demanding. So, I mean, you want to have healthy mitochondria, but you also, I don't know, I think it's a little bit difficult, but I hope we can answer some of those questions someday that you're asking, because it's definitely, I've thought about it too, and I'm definitely interested in, you know, how much too much is. It's probably not stop and go, except for the mitosis part, but DNA mito damage part.
Anybody else with a question? Well, absent another question, I will. So Anna Scott, thank you for a great talk.
Oh, okay.
So yeah, absent any other questions, I will remind people to please fill out this form if you haven't already started to do so. And also, I believe our next talk won't be until January. I think January 11th, if I remember correctly. So you have the whole month of December free. And with that, thank you again for giving this talk, and hopefully we'll be in touch with one another.
Yeah, I'm definitely, I will be emailing you. So anyway, thank you again for inviting me. Thanks for your attention, everybody. So.
Well, everybody have a good day.
All right.